Nirmalya? What's the time? What is the time? Someone give me the time. 12:40. Okay. Is this? Okay. 12:40. Partha, thank you for eating into my class. Do not appreciate it. Right, so shall we begin? Can everyone hear me? Hello? This is not, this is like, I have a... Oh. Okay. Can everyone hear me? Okay. I have notes. So I'd like one of you to tell me when it's 12:15, so that when it's wait, no, 12. Wait, what is it? Yeah, 1:15. Someone tell me when it's 1:15, so that I can know to wrap it up. Yeah. So today I'm going to be talking about the history of science. Can we dim the lights, please? Thank you. I'll be talking about the history of science, and certainly I won't be talking about the entirety of it, because the history of science is as old as civilization itself. So what I'm going to do is I'm going to pick a few examples today, one today and one tomorrow, and I'm going to see if we can derive any general trends that scientific inquiry seems to follow, and hopefully after a week of mathematics and physics and biology and so on, this will be a bit like a storytelling session, which I always enjoy. I never enjoyed this sort of workshop when I was, but these parts I enjoyed. So hopefully some of you are like that. It is an unpopular topic, at least at this institute, in the sense that most people don't think it's worth talking about. It's typically left to the students to. By the way, can everyone understand what I'm saying? Should I speak slower? Everything's good? Okay. Yeah. So this is typically an unpopular topic. It's left to the students because nobody wants to really teach it. And so it's not taught, in fact, in any science course. In fact, Whitehead went as far as to say that any science which has not forgotten its history is dead. Right? What this reflects is a sort of forward-looking philosophy that most sciences have, which is don't look back, look ahead, look at what problems you have to solve. And that is the message that we as graduate students here get, which is forget all that stuff, you got to solve your own problems, right? Which is okay, but the science history need not be a bore, and that's what I'd like to try and convince you of today. So the hope is that maybe if we understand history better, and this is the reason why we learn history in the first place, is that we will get a better understanding of how human beings go about answering questions with experiments and hypotheses, and hopefully give us some hints as to how to try to do it better. So our story begins in 1860s with Friedrich Nietzsche, who's a German biologist and chemist, and he discovered an acidic substance in the cell nucleus, which is somewhere there, and he called it nuclein, right? He called it, sorry? Nuclein. Nuclein, yeah? And so the cell nucleus is somewhere over there. He found an acidic substance. We know what acids are, right? Acids, what are acids? Anyone? Feel free to shout out an answer. Corroding agent. Okay, H plus ions, that's good. That's so everyone knows that, right? Like H plus ions are sort of acidic. That says, yeah. So Miescher discovered an acidic substance in the nucleus, and we call it nuclein. By the way, this talk is about the history of DNA research, in case I didn't already say it. So we start in the 1860s. Now, if you look through Miescher's papers, there is he actually has some comments on heredity, right? Does anyone know what heredity is? Yes. What is heredity? Passing, yeah, so how animals are able to pass on their characteristics to their offspring, right? So I look like my parents, I don't plan to have children, but like if I did, they would look like me, that sort of thing, right? If you look at Miescher's papers, he actually says that heredity is consequent upon the stereo architecture of only a few very large complicated molecules, right? Now when I read this, I was like, wow, this guy knows about DNA. It's amazing. right? Because that's exactly what DNA is. It's a very large and very complicated molecule, right? But you have to be careful when you're looking back into the past and into the papers of these people, right? Very often, it is easy to take a quote out of context, and in most cases, you know, since you know how history played out, right? It's always easy to assume that people were much smarter than they were, right? So, in fact, if you look at his other papers, Miescher was most probably talking about proteins. He was not talking about DNA. We've all heard of DNA, right? Yeah? So he was probably talking about proteins and not DNA itself. So you should always keep that in mind. In hindsight, hindsight always makes people seem much smarter than they were, right? So yeah, let's go back even further. There was something that you're all familiar with. Can anyone tell me who this gentleman is? Mendel, Gregor Mendel, right? He was a friar, which is a strange title, but he was a sort of monk, and he established the laws of inheritance of characteristics. He did experiments with something like 29,000 pea plants, right? And he did very, very careful experiments, and he was able to observe certain laws. So what he was doing is he was looking at characteristics, for example, the color of the seeds, or the color of the flowers, or something like that. He was looking at these general characteristics, and then he crossbred, and then he saw what happened, and he counted up, and he was able to come up with laws. Does anyone know what these laws are? Yeah, go ahead.
So the law of independent inheritance and
Okay.
law of assortment.
Okay, fine. But well, basically, they have very complicated names. The names don't matter so much. The basic ideas are that there is dominance, which is that all characteristics have two contributing factors. One is called a dominant allele, and one is called a recessive allele. Right? We've all heard this. So dominant and recessive. Now, if you have two dominants, then you exhibit the dominant phenotype. If you have a dominant and a recessive, you exhibit dominant phenotype, right? And if you have both recessive phenotype, right? So this is, and you would have seen these like square diagrams where you know, in your textbooks and stuff, you must have seen all of this. So this is all very good science, in fact. It's very, very carefully done experiments. Unfortunately, he published this in a very, like no one had heard of it kind of journal, right? He had published it in a journal that no one had really heard of, and it was ignored for the most part. He also introduced the concept of factors, which are units of heredity, right? And which are really the subject of this entire talk. Now, cut to like 40 years later, Theodore Boveri and Sutton, they came up with their chromosome theory of inheritance. Now, does anyone know what a chromosome is?
Yes.
It's that thing over here. Yeah, it's sort of like DNA, and then you wind it up, right? But basically, what the idea is, if you go back to this image of the cell, you can see over here there's something called a centrosome and the centrioles, right? Now, what happens is during cell division, these guys go to opposite ends of the cell. You must have seen this, right? Like they go to opposite ends of the cell, and then the chromosomes they take this they take this shape, right? Spindles, right? Centromeres, all of that, and then you have these fibers that come down and then break them apart, and so you have two daughter cells, and there's replication and all that. You guys know about this, right? So I won't go too much into the details because this is about history. I'm not trying to teach you. And you must have seen this in Ankit's talk and Tanmay's talk. You must have seen some of this at least. So Sutton and Boveri come up with the chromosome theory of inheritance. And what it does is it basically identifies chromosomes as the carriers of genetic material, right? Chromosomes inside the nucleus as the carriers of genetic material. And what they observed, what you can observe under a microscope, for example, and some of you may have seen slides under a microscope, where chromosomes in cells, they actually split up in a Mendelian fashion, right? And this was, again, a very controversial theory when it first came out, but there was further confirmation by Eleanor Carruthers and Thomas Hunt Morgan, right? Now, Carruthers was a very famous biologist, and she essentially worked on the independent assortment part of Mendel's laws. Independent assortment says that the gene for eye color and hair color, which are presumably different, one does not depend on the other. They will segregate independently, right? So she did some pioneering experiments with grasshoppers, right? and Thomas Hunt Morgan studied mutations in Drosophila melanogaster. Does anyone know what that is? It's a fly. It's a fruit fly, right? And in fact, if you go to biology departments anywhere in the country or in the world, they always have a room full of flies, right? Because flies, they reproduce very fast, and they also have a lot of characteristics that you can test, like eye color, like wing size, and things like that. So, it was because of Thomas Hunt Morgan's work that Drosophila became a standard place to test out ideas, right? So there was further confirmation, and by the 1915, it was established that chromosomes were the basis of genetics and heredity and so on. However, they weren't asking the right question. They were not asking the right question. What they had established was that transfer of complicated molecules is necessary to pass on genes, right? They had established that you needed a big chunky molecule just to break up into two pieces and then go to different like cells, gametes, right? Sex cells. However, the manner in which this copying and transmission was accomplished was never addressed. It was, people just assumed that it happened somehow, right? And so they never considered it important to ask how that happened. They just said it happened somehow, and we're going to just sort of phenomenologically characterize what's going on, right? So they didn't ask the right question. And you can't blame them for this, of course, because like the techniques were very, X-ray diffraction, for example, had just been discovered. 1912, X-ray diffraction was discovered. Sutton Boveri was around 1904, 1905, and then further confirmation came later. But more advanced techniques were just not available, right? But this is going to be a recurring theme, which is that it's very important to ask the right question. You can be looking at the same thing, like 12 people can be looking at the exact same paper or the exact same data, right? But you have to ask the right questions. If you ask the right questions, it might be the case that you will get the answer faster than someone else. So asking the right question has been a sort of a very, very important part of, and it will continue to be an important part of our story. Also, nucleic acids that Miescher discovered. Can we get, Tanmay, can you give me some water? Thanks. So, yeah. Oh, Tanmay. Never mind. So, nucleic acids, which Miescher discovered, were largely ignored. If you look at the publication record, right, from 1900 to 1930, almost no one was talking about nucleic acids. Which is quite surprising, because nucleic acids, as we know today, are important, right? DNA is a nucleic acid, deoxyribonucleic acid, like ribonucleic acid. These are important, but no one was talking about them. One of the reasons is biochemistry was just becoming incredibly powerful as a tool, right? You had all these proteins, right? And you needed to understand their chemical basis, and these had immunological sort of consequences. If you understood proteins, the chemistry of proteins, you could perhaps cure diseases, people thought. So biochemistry was becoming a very important subject. Also, the techniques for isolating and studying nucleic acids were very underdeveloped. So these are a couple of reasons why it may be that nucleic acids were ignored. But perhaps more than anything else, there was what is called the tetranucleotide hypothesis. I don't know if this is taught in school. Is it? Have you seen this before? No. So basically the idea is this. Nucleic acids consist of 4 bases, right? What are the 4 bases? Okay. Okay, so has everyone heard of this? Right? So I mean, if you haven't, it doesn't matter. It just means that like DNA is a big molecule, and it has these 4 nitrogen-based compounds, and they are called A, T, G, and C. They have complicated names, and the names actually have a very interesting. So just as a slight aside, do you know why guanosine is called what it is? Guano is, I think, Spanish for bat poo, right? It's just bat droppings. Guano is Spanish for shit, basically, and so that's where it gets its name. So I mean, again, history, right? It's nice to know this stuff. Anyway, the tetranucleotide hypothesis is basically was put forward by Phoebus Levine, and what it said was that A, T, G, and C, these guys form this tetramer, right? Which is just 4 molecules connected to each other, right? And Levine's experiments led him to conclude erroneously, right? This was wrong. We know it We know that it's wrong today. But he concluded that DNA is just this tetramer, right? This big molecule that consists of 4 parts. It consists of this same unit, but repeated again and again and again, right? Now we know that we know that the chromosome contains 2 parts. It contains DNA and it contains chromosomes. Chromosomes contain DNA and histone proteins, right? Proteins, right? Proteins allow them to fold and things like that. So DNA, chromosomes consist of, this is correct, right? Yeah. So just correct me if I'm, because, yeah. So chromosomes consist of DNA and proteins, right? So what Levine thought was that DNA formed the backbone and the actual genetic material was carried by proteins. Now we know that DNA is responsible. We know that whatever these genes are, they are responsible for variety, and we see a lot of variety even among human beings. Right? There's a lot of variety. So Levine concluded that something as simple as this repeating units of this big molecule cannot be responsible for the genetic code. So he said nucleic acids are not important. Nucleic acids are just a backbone. So this was called the tetranucleotide hypothesis, and basically what he says is that nucleic acids are too simple to carry genetic information, right? So this is at this point there is this is relevant. Dogma and authority, as much as we would like them not to be there in science, they are very much a part of it, right? Very often incorrect hypotheses have been accepted, sometimes solely on authority. This has happened again and again in the history of science, right? And there are plenty of examples, and I urge you to find more, right? And in fact, the tetranucleotide hypothesis took until 1952 to be disproved, right? This was made in the early, like in the first 20 years of the 20th century. So it took a long time, right? So dogma and authority are parts of science, unfortunately, and we'll try and speculate as to why that is the case later on in the talk. Now we come to Griffith's experiment, and you must have all heard of this. Can you see the diagram? Yeah. So what you have essentially here is that you have these S cells and R cells, right? Now the only difference between S cells and R cells are that S cells have this protective coating of sugar, right? Or if you want to sound fancy, you can say polysaccharide, but it's essentially sugar, right? Now S cells have a protective coating of sugar, and R cells do not. Which means when you put S cells into a mouse and you put R cells into a mouse, then the mouse's immune system can fight off the R cells because it doesn't have a protective coating, but it cannot fight off the S cells because it has the protective coating, right? So the mouse dies when you inject live S cells into the mouse, and it lives when you inject R cells, right? So Griffiths did this wonderful experiment where he heat killed the S cells. He just burnt them, right? And he injected them into the mouse, and he found that the mouse was healthy, right? And then what he did was he put a mixture of heat-killed S cells and living R cells, right? And he injected this into the mouse, and the mouse died. Sorry? It should have survived, but it died. Because R cells don't do anything.
R cells replace the S cells.
R cells do not replace the S cells; they become S cells. No. So can we have guesses? Anyone would like to try?
They form a coating around them, like sugar coating around them.
Yeah. So what is happening? So clearly there is something inside this heat-killed S cells that is fundamentally altering the nature of the R cells, right? That's what's happening. It's very obvious that is what is happening in this experiment. Now, what is that?
Maybe their genes are not killed.
Yeah. See. So that's a good idea actually, and that's in fact what he thought. And in fact, when he did an autopsy, is it called an autopsy when it's an animal? Okay, because humans have autopsies. Anyway, when he looked at the cells from the dead mouse, this is sad. He found that there were living S cells. Right. So what's happening is something from the heat-killed S cells is going into the R cells and changing them, right? And after that, they're becoming S cells in the next generation. Which was a big deal at the time, because earlier work by people like Wilson, they were pretty sure that if you had different types of bacteria, they could not transform into one another, right? So this is called transformation, and bacteria can transfer genetic information, which is basically the message of this experiment. Now, the big question is, of course, is the transforming principle DNA, RNA, or protein? right? Because by that time we knew that these were the only 3 molecules that were complicated enough to do the job. Was it DNA, RNA, or protein? So what Avery, McLeod, and McCarthy, this must also be in your textbooks actually, it's a very famous experiment. What they did was, they killed the S cells, right? And they removed any lipids and carbohydrates that they could find, right? So the only thing in this beaker over here is DNA, RNA, and protein, right? Then what they did is, they separated it into 3 flasks, three beakers, and what they did is, they added compounds that would destroy proteins in one, they added compounds that would destroy RNA in the other, and in the third one, they added compounds that would destroy only DNA, right? So you understand what's happening here? By a process of elimination, right, we're trying to find out which one it is. It's a very, very clever thing, and in fact, you cannot do this if you didn't understand their chemistry very well, which was already known by then. So you add compounds that destroy each of them individually, but leave the other 2, right? And then you add the R cells, and then you see if transformation happens. And it was found that when you destroyed the protein and you destroyed the RNA, transformation still occurs. But when you destroy the DNA, transformation no longer occurs. Very good. See, I mean, you could have thought of this experiment, right? It's very simple. I mean, of course, the techniques are slightly complicated, or but the idea is very simple, right? It's just a systematic elimination of all the possibilities, and they found out that DNA was the one. It was still a fairly controversial experiment until Hershey and Chase did another very beautiful experiment. This is Hershey and this is Chase. Is it the other way around? I don't. The other way around. Okay, sorry. Yeah, these are Hershey and Chase, so they were part of the what is called the phage group, right? These guys worked on a set of viruses that were called bacteriophages. Sorry? They harm bacteria, right? That's where the name bacteriophage comes from, right? So these are viruses that harm bacteria, and they have a protein coating and DNA inside, right? So what they did was, what did they do in this experiment? They tagged, okay. What they did was, they know that the protein coat has certain, do you know the details of this? Something happens, right? So they tag the DNA they tag the nitrogen in the DNA and the sulfur in the coating, right? So no, sulfur is not in DNA, it's in protein. No, they tag the nitrogen in the DNA and the sulfur in the protein. Nitrogen in the? That's what I said. Okay. So what they do is, do you know what tagging is? What you do is, so you let these guys grow in a bath, right? That is filled, that's nitrogen rich, except it's not rich in ordinary nitrogen, it's rich in an isotope of nitrogen. Do you know what an isotope is? Yes. What is an isotope? Yeah, that sounds about right. So you grow it in in a bath, and you can separate these because one is heavier than the other, right? So you can separate these using various techniques. Now, what you do is you tag the sulfur in the protein coat, and you tag the nitrogen in the DNA, and you let it infect cells, right? And then what you do is you remove the capsules, right? You can do this because they have different weights. So you remove the capsules, and then once you centrifuge, you will see that they were able to establish that it was not the proteins but the DNA itself that that carries genetic information and that is capable of changing the cells and so on. Now we get to the hot part of the story, right? This is the story of high drama. It's in fact one of the most exciting discoveries of the 20th century. This is the structure of DNA. Right? And we have 3 groups of people working here. We have, this is? Watson and James Watson and Francis Crick, right? These guys were at Cambridge. This is Wilkins and this is Franklin, right? They were, sorry? Pauling, oh very good, okay. Yeah, so Wilkins and Franklin were at King's College, London, and Pauling was at Caltech. Now these 3 groups decided they were going to be the ones to do it. So what happens next is absolutely extraordinary. Pauling had a few years earlier established that proteins can fold themselves into what is called an alpha helix. Now a protein is a chain of amino acids, right? We know this. Now what is an amino acid? An amino acid is something that has a carboxyl group and an amine group, right? Like a C double bond O and an NH2. That's what an amino acid is. And there is some R, some other group. It can be a methyl group or whatever. It can be a whole number of things, right? And there are a whole bunch of amino acids that you can get as a result. Now, what he showed was that the amino acids can fold themselves in the following manner, right? And that there are hydrogen bonds that stabilize this structure and so on. So Pauling had discovered the alpha helix, and X-ray diffraction, X-ray diffraction data that Wilkins and Franklin were able to get also suggested that there was a helical structure, and this is called photograph 51. It's a very famous photograph. This was the one that led Watson and Crick to figure out that it was a helix and that it had certain properties and so on and so forth. A very famous photograph, actually. So they knew that helices were important, but
Why is it called photograph 51?
I don't know. Just like I have no idea actually. Like why not, right? I mean, maybe it was 1951. Yeah, it was. Yeah, they found it in 1951. It's not a very interesting answer though. It's kind of boring. But anyway, it's an extraordinary photograph. So do you know what X-ray diffraction is? No. So X-ray diffraction is very quickly, right? X-ray diffraction is if you shine light against something and you look at the reflection, usually you'll be able to make out what the object looks like. Now, that's very vague. That's very, very vague. But what you can do in the case of like a crystal structure, for example, is you know that waves can sometimes add up and waves can sometimes cancel out, right? We know this. So what happens with the case of X-rays is that sometimes they will add up and they'll give you these white spots, right? And sometimes they'll cancel out and give you dark spots. So there is a theory in physics and uses a lot of mathematics. Sorry, yeah, you wanna? Sorry? No, go ahead. I mean, yeah. Well, Young's double slit experiment is an example of diffraction, but what we're talking about here is crystallography. So how do you know that a certain compound has a certain crystal structure, right? And how you determine this is by looking at plots like this and then trying to see trying to work backwards and say, if this is the image that I see, what possible structure could have produced this image, right? And there is a very elaborate theory that explains how this is done, and which you all learn, which you will all learn if you continue to do physics, for example. Now, so this X-ray diffraction allowed us to determine that there was a helix involved, but there was also a lot of drama at this time. First, Crick and Stokes, they had worked out what the pattern would look like for a helix, what the diffraction pattern, or let's just call it photograph, right? Diffraction pattern is an awkward, long word, not quite sure what it means. So let's just call it a photograph. So if you take a photograph of a helix, Crick and his collaborators had figured out what the photograph would look like, right? At the same time, the guys in the UK, right, which is Franklin, Wilkins, Watson, and Crick, they wanted to beat Pauling because Pauling was already famous. He already had his Nobel Prize, and he had figured out the alpha helix, and they thought he was also working on the structure of DNA, and they wanted to beat him. Right? Naturally, competition is sort of an important part of this game. So they were competing with Pauling, and in fact, Pauling was in I think 52 or something like that. He was scheduled to come visit the United States. Come visit the United Kingdom. And if he had, but what happened was he was politically very active, and the United States government did not want one of their most famous scientists going around saying all kinds of stuff. So they canceled his passport and all this drama. But basically what happened was he was not able to go. And as a result, he did not find out that these two groups in the UK were working on this problem, right? So God knows how things would have happened, right? If Pauling had come, he had and he had gone to those labs and he said, "What are you working on?" They would have had to say, you know, we're working on DNA structure and this is what we think. And I mean, Pauling was the greatest chemist alive at the time. So if he had known what they knew, he probably could have figured it out himself. But the trip was cancelled because of political reasons. So that was not possible. So the guys in the UK, especially Watson and Crick, they were in such a hurry to beat Pauling that they had one attempt, and it was a huge failure. And the reason it was a huge failure was because they didn't know as much chemistry as Rosalind Franklin. And Rosalind Franklin, so they actually went to her with a structure. So let me just skip ahead. And so we know that this is what DNA looks like, right? We've all seen this in our textbooks. We know that the bases are inside, and the phosphate sugar forms like a backbone. right? The first guess that Watson and Crick had was that the bases were outside and the phosphate sugars were inside, right? But here there's a very simple problem here, which is that phosphate groups are which one? Hydrophobic or hydrophilic? It's ionic, okay? The phosphate group is ionic, so it will like water. It will be hydrophilic, right? Whereas these nitrogen bases, they will be hydrophobic. Does everyone know what these words mean? No? Someone doesn't know. Okay, so hydrophilic means I like water and I want a lot of water molecules around me. And hydrophobic means I'm afraid of water and I don't want a lot of water molecules around me. Okay? So that was their original idea. And what Franklin said, so they suggested this idea to Rosalind Franklin, and she just said, look, if you put this in water, it's not going to, there's no way the structure is going to be stable. Because you have all the guys who are afraid of water outside, and all the guys who like water inside. It's not going to work, right? So Franklin's knowledge of chemistry actually helped a lot in clarifying the actual structure. Then came along Chargaff's rules. I don't know if have you heard of these? Chargaff's rules basically say that the amount of A and T is the same, and the amount of G and C is the same, right? So the ratio of A to T and G to C, these are nucleotides. These ratios are the same, 1:1, always, right? This is called Chargaff's rule, and Chargaff's rule essentially, you can realize that like what this rule would do is it would invalidate Levine's tetranucleotide hypothesis, right? Because all it says is that A's to T is 1:1 and G's to C is 1:1. But they don't say that all four have to be 25%, 25% each, right? And in fact, if you look at like lower animals, like bacteria or things like that, then they do have very skewed ratios. For example, so these are called purines and pyrimidines. So the ratio of the purines to the pyrimidines is very skewed in smaller organisms, but it's roughly equal in larger mammals. So another thing that helped was the use of molecular models, right? Pauling was already an expert at doing this. This is how he had arrived at the alpha helix. That's correct. This is how he had arrived at the alpha. In fact, there's a video on YouTube where Pauling is drawing this chain of amino acids, and then he says, "This is how I figured it out." And then he just folds the paper, and it becomes a helix, right? So it's a wonderful video. You should check it out. It's on YouTube. What happened was, I think this was late '52. Pauling and Corey, they had written a paper with an incorrect proposal for what the structure of DNA was, right? And this incorrect proposal, Watson got his hands on a preprint, right? So Watson found out that they had made this incorrect proposal, and what he did was he ran all the way to London, and he said, I mean, he didn't actually run, he maybe walked, but he went all the way to London, and he tried to convince Franklin that they should all work together, because if they don't, then Pauling is going to find out the mistake in his paper, and he's going to correct it, and he's going to figure it out, right? So there's all this stuff happening, right? But of course, Franklin was a very proud woman. She was very, very intelligent and very proud woman. What she understood when Watson said this was, she thought Watson was saying that she didn't know how to interpret her own data. So she got insulted, and she asked him to leave. So Watson was leaving, and when he was leaving, he runs into Wilkins. And Wilkins reads the preprint, and he realizes that, Pauling is almost there, right? So what he does is he shows Rosalind Franklin's photograph 51 to Watson without her knowledge. And this sort of changed history, right? Like, it's an absolutely chilling moment if you think about it. This, more than anything, led Watson and Crick onto the right model for DNA, right? And there has been a lot of controversy surrounding this. But I'll come to that at the end. There was also a huge clash of personalities. What I like about this story is that there is a huge clash of personalities involved, right? In The Double Helix, which is a wonderful book, and I recommend all of you take a look at it, because it's a very personal account of what happened in those few years. Watson actually says things like, in contrast to the popular conception supported by newspapers and the mothers of scientists, a goodly number of scientists are not only narrow-minded and dull, but also just stupid. This is the kind of things he said, and this upset a lot of people, right? But what I'm trying to illustrate with these examples is that our science is objective, right? The truths we find are objective. They are true independent of who discovered them or what his name is or how rich he is and so on. But the people who do science are not objective. They're very subjective. Like any other group of people, right? They're just as susceptible to prejudice and anger and jealousy and so on, right? And this has, like over the course of history, has changed the course of history a number of times. And like there will be more examples hopefully tomorrow. There were also other clashes of personalities. Wilkins and Franklin, although they were working in the same lab, they just did not get along because Wilkins was a very shy man and he was very quiet, and Franklin was very abrasive, and she just looked people in the eye and she talks to up to the point where it's just like she's being rude, right? She was brilliant, absolutely brilliant. Both of them were, but they just had different personalities. Crick also managed to upset Lawrence Bragg, who was the director of the place where Crick worked. So he managed to upset him a bunch of times, and that didn't help because at one point Bragg said, "Stop all this DNA nonsense, get back to your PhD." Because at the time Crick was 35 years old and he didn't have his PhD. So like people were getting a little upset, right? So there are all these clashes of personality, but in the end they figured it out, and it's a nice thing to know. They figured it out that there is a double helix, and that the sugars and the phosphates, they form a backbone, and that the A and the T always combine, and the G and the C always combine, and that the nucleosides are on the inside, and so on and so forth. Right? These were later confirmed by some. This is called one of the most beautiful experiments in biology. I don't know. Have you seen this? The Mendelssohn-Stahl experiment. You probably will. The basic idea is now DNA has to replicate. Right? Now this is two strands. Right? They're intertwined, and it has to replicate. So the strands have to unwind, and then they have to, you have to sort of use one as a template to construct another strand, and so on, right? This was called semi-conservative, right? And there are some technical details. I don't think now would be the best time to go about it. But what I would like to impress upon you is that these are very simple experiments. Not simple in the sense of actually carrying them out, but the idea is very clear, right? There are like one or two basic principles at hand. And you just have to be clever enough to think of it. And what they did was, in fact, and when you read about this, and perhaps we can talk about it over lunch if you'd like, if you're interested. Very simple experiments that show conclusively, right, that such and such is true, that semi-conservative replication is true. And later on, the central dogma of molecular biology was established, which is that DNA, and I think Tanmay, you'd spoken about this, right? Yeah, so DNA transcribes, gives you mRNA, and mRNA is translated to give you proteins, right? This was established, and this was also verified by Crick and his collaborators. So it's not just discovery of DNA, right? There was a whole lot of work because these guys were just suggesting an idea, right? Now you had to go and confirm that idea. So there were a slew of experiments done, and all of them, without exception, came back and said, yeah, this is the right model. This is exactly what we need, right? Yeah, there are, okay, maybe we'll do that. So what conclusions can we, I think my time is almost done. It's done, actually. So what conclusions can we come to at this point about science? And I'd like to say more about this tomorrow. The first is that it is an international activity, more so today than it was then. Back then you had people from all over continental Europe working together, and you had them competing with the Americans, and there's all this stuff going on. But today it's a more collaborative activity. There is competition, but like you see, for example, you must have heard of the Large Hadron Collider. It's this big particle accelerator in Geneva. Yeah, yeah. Some of you have heard of it. It's an impossibly large-scale experiment. Huge number of people. Something, yeah. Well, I don't like saying that, but okay. Now, so it is a large-scale activity. You have a huge number of scientists all working on the same thing, and it's amazing that they're able to figure out how to let everyone talk. You know, that itself must be a huge logistic nightmare, right? The other thing is inference from data. You pay careful attention to what the data is telling you. Don't go off thinking about some. So this is how modern science is differentiated from, say, the science the way Aristotle did it. Aristotle said flies have 4 legs, and then no one bothered to check. Flies don't have 4 legs, right? Yeah, they have 6 legs. But Aristotle just said they have 4 legs, and because Aristotle was Aristotle, everyone said, okay, must be right. And they just went along, right? No one checked. So you have to pay attention to what nature is telling you. That's the second thing. The third thing, which I will stress on tomorrow, is experimentally verifiable hypotheses. So you make guesses that can be tested. I remember when I was in school, I didn't like going to school, so I used to tell my mother I had a headache. This is not experimentally verifiable. No one in the world can tell you whether you're having a headache or not, right? At least not at home. So this sort of thing doesn't fly. If you say you have a fever, then your mother can take your temperature, and if you're don't have a fever, then she'll slap you and send you to school. But experimentally verifiable hypothesis is very important, right? And also the importance of engineering and technological developments. Absolutely essential, right? If you don't have engineers figuring out how to make measurement apparatuses even better and so on, things are not going to work out, right? The second set of conclusions, this is my last slide, so don't get too nervous. It's important to ask the right questions. Absolutely essential to ask the right questions. It's not clear what the right questions are, right? Which is why it's like doing science today is nice, because you have a lot of people bouncing around a lot of ideas. Statistically, one of them is likely to be right? So the second thing is that dogma and authority have significantly influenced scientific progress. This is a truth. We know it. We've seen lots of examples of it. The third is that it is a human activity. So prejudices, bias, clash of personalities, important pieces of work being neglected. This happens all the time, right? And I don't see any way to sort of get around it, but that's just how it is. And the fourth is criticism, how important it is, right? How important it is to be able to listen to someone say, no, you're wrong, that's just incorrect. And just say, okay, fine, it's wrong, let me move on to the next thing. So tomorrow, hopefully, I'll be able to talk a little bit more about the scientific method and what it is and so on. Thank you. Does anyone have any questions, or are you hungry so you'd like to leave? Prefer to leave, okay. Yeah, fine. If you have any questions. So I think I will be preparing a set of notes that have a lot more. Kamal Akshar will put them up on his webpage. So you can take a look at those. Those have references, so you can follow them. And if you're interested, you can write to me and we can have a discussion, that sort of thing. Thank you.